Hello, my name is Tarek Obeid. I am a second-year medical student here at the UCF College of Medicine, and welcome to my presentation on elucidating the role of dopamine receptor D4 in cartilage. To begin, it's important to note that osteoarthritis is a widespread and debilitating disease that affects articular cartilage, and human chondrocytes are the cells responsible for creating that articular cartilage, as well as secreting type 2 collagen. There are current therapies for osteoarthritis. However, they are primarily palliative in nature, and they don't have the regenerative capabilities that we want them to have. So there's a strong need to identify novel therapeutics in order to prevent osteoarthritis or the progression of osteoarthritis. A recent natural product screen, aromoline, which is a bisphenzyl quinolone alkaloid, was identified as being able to stimulate type 2 collagen expression. It also showed upregulation of its target protein, dopamine receptor D4, also known as DRD4, along with type 2 collagen. The identification of DRD4 in cartilage was a novel finding, but its role is unknown. CRISPR-Cas9 was used in our study to knock out DRD4 and observe its role in chondrogenesis. That leads us to our objective: investigate the role of DRD4 in chondrogenesis using primary human chondrocytes and the chondrocyte cell line TC28A2. Moving on to our methods portion. Immortalized human chondrocytes and primary human chondrocyte isolated from a 4-day neonatal femur were cultured and maintained. In order to confirm the expression of DRD4 in these cells, they are subjected to immunocytochemistry and qPCR. The immunostaining was carried out using an anti-DRD4 monoclonal antibody and hushed staining for the nucleus. Fluorescence microscopy was used to visualize the cells, and to validate the presence of DRD4 at the RNA level, qPCR was performed with hPRT as the reference gene. A lentiviral vector, lenti-CRISPR-V2, was used for knockout experiments with a guide targeting DRD4 and a negative guide for the control plasmid. Lentiviral particles were applied to the cells with polybrine. The growth media was replaced with media with puromycin in order to select the infected cells. The DRD4 knockout of IAM and TC28A2 was subjected to immunocytochemistry in order to confirm the loss of DRD4, and qPCR was performed in order to verify that knockout. The evaluation of chondrogenesis was done by having a TGF-beta dose response on both IAM and TC28A2, and they were analyzed over a 21-day period. 3D aggregates of IAM TC28A2 cells were imaged, and a pellet cross-sectional area was determined using ImageJ software. In our bottom right corner, we have our proposed interaction of DRD4 with TGF-beta, with aromoline binding to our DRD4 receptor, leading to downstream mechanism of action, along with TGF-beta and its downstream mechanism of action, both leading to the expression of type 2 collagen. Moving on to our results portion. Here in figure 2, we can see our qPCR confirmation of DRD4 knockout of both IAM and TC28A2 cells. Over here, we see both control cells had a cycle threshold of about 40 cycles, and both the knockout cells did not have any detection after about 45 cycles. Moving on to our immunocytochemistry, we have both our IAM and TC28A2 cells. And this is where we see our anti-DRD4 monoclonal antibody stained in red, and our hush staining for the nucleus in blue. The blue representing the nucleus of the cell. We can see in both the untreated controls of TC28A2 and IAM, both of the cells are present, and the DRD4 stained in red shows DRD4 is also present. Moving on to our plasmid control, same thing as the untreated, we see our cells present in blue and the DRD4 staining in red. In our DRD4 knockout, we see its cells stained in blue in both in both cells. However, we do not see red, confirming our DRD4 knockout. Moving on to figure 4, we have our TGF-beta dose response to IAM cells. In pink, we have our control, and black, we have our DRD4 knockout, with an EC50 of 1.914 in our control and EC50 of 2.531. This is basically just telling us that there's a decreased sensitivity to TGF-beta, which kind of aligns with our previous thinking that DRD4 is modifying the TGF-beta dose response. Down in figure 5, we have our TC28A2 cells, which showed, interestingly, a flat response to the TGF-beta dose response in both the DRD4 knockout as well as the control. This is basically suggesting that the immortalized cell line is a poor model in this case. Moving on to our discussion portion. The study investigated the role of dopamine receptor D4 in chondrogenesis, identifying it as a novel G protein expressed in human chondrocytes and establishing its connection in TGF-beta. The successful knockout of DRD4 strengthens it as a potential therapeutic target in aromoline's mechanism of action, enhancing type 2 collagen expression. The discovery represents a significant breakthrough, recognizing DRD4 as a promising therapeutic agent for osteoarthritis and hopefully filling a critical gap in available treatments. The data advocates for further investigations to delve into the intricate signaling pathways and molecular details associated with DRD4 and chondrogenesis. And with that, we conclude our presentation. Thank you so much for joining us.